Thank you. Okay, so I'm going to talk to you a bit about looking at adolescence in bioarchaeology. So why is this an important aspect of bioarchaeology? Well, adolescence is a unique stage of the human life cycle. You have encompassing things like the growth spurt, sexual maturation, known as puberty, and also brain development, which is something that I think as biologists are very interested in the physical body, we forget. It's a major area of research in human bioarchaeology and psychology, neurobiology and medicine. There are a mountain of research out there on the psychological changes in adolescence and the biological changes in adolescence. But it's something that we tend not to really look at in bioarchaeology. But it's very much understood that adolescence is very complex and dynamic, both in physical changes, but also psychological changes. The way that you're treated or the way you feel about being an adolescent, or even how long it takes you to be an adolescent, how long you're an adolescent for, is culturally defined. And also it's a very individual experience. So the WHO, that's the World Health Organization, define a young person or an adolescent as someone aged between 10 to 24 years of age. And so this might seem quite old for us to have a 24-year-old still considered a young person or a vulnerable child, but they are, because they are still developing psychologically and are still very vulnerable. So any research into adolescence encompasses that age group. So as I said, it's not something that we've explored that much in bioarchaeology, and I think it's because of small sample sizes. Teenagers tend not to die. But I'm going to talk to you about how we can still get a lot of information out of these adolescent skeletons, even if you only have one or two individuals. So I'm going to use teen as a shorthand, because obviously we're going out beyond the teenage years. But the biological teen, if you consider what's happening to an adolescent biologically, puberty, which is the sexual maturation, is part of adolescence. It's not the whole of adolescence. But there are both hidden changes that start within the brain first with hormone releases, but also overt changes, changes that people can see around you. And this affects the way that an adolescent behaves and the way that they're treated in their environment. So the adolescent period encompasses the growth spurt, where an individual will put on about 50% of their adult body weight and 20% of their stature. So it's quite crucial, and particularly nutrition is quite crucial during this period of time. So you can study things that are changing in the skeleton, from bone mass to somatic growth, so linear growth, skeletal maturation, and skeletal morphology. All of these things change, and we can see them visibly on the skeleton and measure these things on the skeleton. But also in an adolescent, their immune system is transitioning. It's going from a child's immune system that tends to contain a disease to an immune system that starts to attack a disease. And so we see new diseases appearing, or harbored diseases starting to appear again in the individual, which makes paleopathologists, and I confess that I am at my heart a paleopathologist, it makes paleopathology of adolescence quite an exciting area to research as well. But from the point of view of the society, when you've gone through adolescence, towards the end of adolescence, you're capable of adult labour. So after the peak height velocity, the peak of your growth spurt, males put on their musculature. So you can see here, Hiccup has gone from being a child to an adolescent. He's much taller and much more muscular than he was. So the way that people may have treated adolescent society would have changed depending on where they were on the growth spurt and how they appeared externally to that society. But also women would become capable of getting pregnant, they may be getting married, but certainly there's a change of identity that comes with being an adolescent going on into adulthood. So as I said at the beginning, it's not just about the physical changes from the body or from the skeleton that we might want to look at. You have to remember that a lot about being an adolescent or being a teenager is to do with your brain. So the brain starts to develop, it initiates puberty, it releases all the hormones that stimulate puberty, and we'll talk about this in the final session. But it also spans a whole of adolescence and goes beyond adolescence, and maturity in the brain continues on beyond the physical changes. what's happening is the limbic system within the brain, the pleasure center, develops before the frontal cortex, which is for rational thought. So this is why you get teenagers are defined as being quite irrational. They take risks, so they're more likely to be exposed to violence or have accidents because of risky behavior. They're into experimentation, so they may experiment with drugs and sex. They're very vulnerable, particularly mentally. They have insecurities, comparing themselves to their peers. They're more likely to commit suicide and have bad thoughts and depression about themselves. And there are particular mental illnesses that we know develop during adolescence, for instance, schizophrenia and obsessive compulsive disorder and anorexia. All of these quite severe mental illnesses occur during adolescence. And so there's an awful lot of research about the adolescent brain that is really interesting if you get the time to look into it. But what you want to take away from this is that the pleasure center is developing before rational thought or control center develops, hence the behavior in an adolescent. But also this long time period that the brain takes to develop means that they're much more likely to be susceptible to stress that might lead to long-term depression in adulthood, but also physiologically more likely to experience stress and have detrimental effects of stress, given the amount of growth that's going on in this period. But quite importantly, what's going on physically is the sexual maturation, the ability to reproduce, is occurring before a lot of the psychological maturation occurs. And this has been looked at by evolutionary biologist Barry Bogan. Talks a lot about this hypothesis that was first talked about by Gluckman and Hansen, their psychologists interested in adolescence. That one of the reasons that you see a long period of adolescence, that this increases in time, the more complex a society becomes, the longer it takes for that individual to develop the psychosocial skills to adapt to that environment and to become and behave like an adult. So in the blue on this schematic graph, this is all hypothetical, it's never been tested. You have quite close in the hunter-gatherer populations, you have quite a close relationship between the physical development and the psychological development. But as we go through complexities of society, there becomes this mismatch between the physical ability to reproduce and be an adult physically and actually behaving as an adult. So this is something hasn't really been looked at in archaeology and bioarchaeology, but I think something we could actually explore in the future. So also we have a way into looking at life history theory, particularly through looking at changes in puberty. So it's a critical window of human development. Timing, the onset of puberty, when it begins, when the hormones are released, is related to conditions that a fetus can experience in the womb. This is known as fetal programming. So it's a mixture of what's happening when the child is in the womb and programming that's occurring at that point, but also in what's going on in their immediate environment and the environment that they're living in, how conducive it is to growth, what their nutrition is like, what that's like just as they hit puberty. So you've got these two things going on. So fetal programming, or this life history theory, is explained that if you're being born into a high mortality population where the chances of you living enough to reproduce are reduced, you tend to hit puberty much earlier so that you can reproduce earlier before your obvious demise. And so this produces what's called an energy trade-off. So this kind of growth that you're experiencing, the growth spurt, is halted in order for puberty to take over and for you to be able to reproduce. So this energy trade-off usually means that, and this is something that Sharon DeWitt and I have recently explored in a paper, that you are more likely to, with early menarche, your first period, the earlier you have your period, the more likely you are to be shorter, because reproduction has overtaken growth. So that's exploring life history theory and energy trade-offs. And menarche itself is very often explored. It's a really good indicator for biologists to explore puberty in the past, but it's also very fragile. So as I said, it's determined by fetal programming, by the conditions you experience in the womb, but also by the experiences of stress and your nutrition. So we know that body weight will affect whether a girl has her period or not. as well as psychosocial problems, so psychological stress, such as your father absence or exposure to violence. All of these things can affect the age at menarche. So it's a really sensitive indicator. And if we, as bioarchaeologists, can get to that, we have a great potential for exploring new avenues in bioarchaeology. It's not going to be easy, though. So I will talk to you about why it's not so easy in the third lecture. But the other thing that I think is really interesting is that adolescents have a really crucial period for future generational health. So young mothers today are most at risk for pregnancy hazards. This is a lot to do with the culture, so it might be something that's a modern phenomenon in that they're more likely to have unstable relationships. They haven't had time to develop stable relationships before they have babies. They're experiencing risky behaviours sexually, so they're more likely to have sexually transmitted diseases. They might be smoking and drinking as part of this kind of pleasure-seeking response in their brain. But also the pelvis continues to develop after menarche, which means even that you have this delay period between having your period and being able to conceive a child. And this is probably because the body's waiting for the pelvis to be the right size in order to allow pregnancy to be successful. So because they're still growing and women have greater nutritional requirements, young women have greater nutritional requirements during pregnancy, which means their children are more likely to be exposed to neural tube defects or have anemia, for instance. We know that maternal nutrition affects the health and survival of the first child, and the weight of the mother when she has her first pregnancy goes on to influence the health of their second child. So young adolescent females are key to the health of future generations and feed into bioarchaeologists' interest in DOHAD, developmental origins of health and disease. So adolescent bioarchaeology, I think, is lots of areas that we can explore if we understand the background to adolescence. So looking at their kind of social behaviour and their treatment, when do they start work and how does that affect their bodies? They're testing out their independence, whether social complexity expands adolescence. There's also problems with obstetric hazards with young females, and also how exposure to stress and violence might affect the timing and tempo of puberty. So a lot of this was explored initially during a project that I did that was a Leverhulme Trust funded project with various colleagues like Janet Montgomery in Durham and Fiona Shapland, who was my research postdoctoral research associate during this research. And we looked at medieval adolescence, and during this research we were able to, because we had such a large number of medieval adolescents, we had, I think it was, we had in our total data set, we had about 3,000, but we had 900 where we could look at puberty. And so we devised these puberty methods. So Fiona Shapland did a lot of work in looking at clinical literature for how clinicians have been mapping skeletal changes with the growth spurt to indicate puberty and where they were along the growth spurt. So we can identify particularly crucial periods. So menarche, when onset of menarche occurs, and also peak height velocity, when they're at the very peak of their maximum growth and body weight and bone mass, which is quite a crucial period of time for health in particular. Okay, so what we did was map these skeletal changes, and I'll talk to you about how we apply this method in the third lecture. But we map these physical changes onto the different stages that we see in the growth spurt. So you have the onset of puberty, when puberty first starts and hormones are released. And this is where the gonads, so the ovaries and testes, start to be enlarged, hormones start to be produced, kids start to get acne kind of thing. Then you have the acceleration of puberty, where you start seeing the sexual characteristics, physical changes start to occur, but this is happening after puberty has actually started. Then we have peak height velocity, where you're getting changes in voice for males and musculature, so they're starting to appear more adult-like. And then we start to go through deceleration as they start to finish their growth spurt, where menstruation and ovulation towards the end of growth, and maturation, where ovulation starts to become more regular and more likely to become pregnant. And you look like an adult, like a full adult. And then you have completion where all of the bones are fused and the growth spurt has stopped. Although development of the brain is continuing slightly longer than that. So part of the research we're looking at these medieval adolescents and drawing on these new puberty methods was to actually explore as a group what ages that people were attaining puberty in this period. So we estimated that the age of onset for medieval adolescents was 10 to 13 years of age, and that the average age of menarche was 15, which is later than today. So it's around 13 for girls today, 12 and a half, 13. Whereas in London, which was quite an urbanized centre at the time, the average age was 2 years later, so girls were about 17. This is suggesting there's some stress in the environment. That's meaning that they're delayed compared to other medieval girls. But one of the reasons that I think that the puberty method works is that we had both early and late developers. So just as we do in modern populations, we have people who are ahead of everybody else and people who are behind everybody else. So the normal variability in puberty development is there in the skeletal record. But also physical adolescence. So people were still developing after 25 years of age, and that's when our data set ended. But then we still had some individuals who had not reached completion of puberty, particularly in London at this time, raising questions about how do we define an adolescent, how do we define an adult in the past. And also what we saw was that chronic conditions, pathological conditions, were delaying puberty. So we explored this in another paper looking at the influence of chronic conditions on maturation. We've known for a long time that skeletal maturation is affected by pathology, but by looking at puberty markers, we've actually got an opportunity to start mapping this much more closely. So of my 900 adolescents, I had about 600 that we could provide a puberty stage for. And 40% of these who were in the pathology group had delayed pathology. So if you look at the slide here, we have 40% of individuals were delayed and they showed some sign of pathology. So it's metabolic disease, infectious disease, or a severe congenital condition. Compared to only 20% who were delayed who didn't show anything on the skeleton to suggest why they should be delayed. And this was statistically significant that the pathology group were delayed compared to the non-pathology group. And this delay increased as you went through the different puberty stages. So these are the different puberty stages. And you see the delay, the red bar, is increasing as you get towards completion. So it's taking much longer for them to get through puberty if they have a pathology than if they don't have a pathology. And in particular, the male skeletons that we looked at were delayed at peak height velocities. That's at the third right here, much more. And that's something that they see clinically today. So boys with severe pathology tend to take much longer to hit peak growth than their counterparts who aren't suffering any illnesses. But more interestingly, there are 111 individuals in a sample who were delayed with pathology and continued to be delayed as they went through puberty, but they didn't have any signs of pathology. So there are probably hidden factors such as violence, exposure to violence, stress, pollution, all things that would have been around in medieval urban society that were really causing delay in these individuals, much beyond what you might expect through normal variation. So I think, again, if we can start mapping puberty and mapping the growth spurt in this way, we can start unpicking some of these factors in the past. One of the other factors that we saw, I'll just kind of share with you, I wasn't expecting, was that of the individuals that we could sex, the adolescent females were the ones that had the most severe respiratory diseases in particular. So the females in this are in the white bar. So they had over double the amount of tuberculosis and sinusitis, maxillary sinusitis, is that you've got air pollution, you're breathing in poor air, and also treponemal disease. So some of the females, it's only small numbers, about 5 of them, had venereal syphilis. What we think at this time is venereal syphilis rather than one of the other treponemal diseases. But they weren't delayed in their pathology or in their puberty. So I suspect that they may, we don't necessarily know that they were advanced, we just know that they weren't delayed. And so this small group of individuals may have been they were exposed to sexual violence, certainly they were having sex at this time, whether that was something to do with their occupation. Were they prostitutes working in medieval towns? We don't know. So I think there's another area to explore there, looking at adolescent females in particular, and remembering back to what I said about how important the health of females is during this period of time for the health of the next generation, for having children. Okay, so since we published the method, many people have started to explore this. So far, most of the research, in fact, all of the published research has been done in Europe. So if you're listening from North America or Australia, then here's your chance to get in on this. So few people have looked at American populations, for instance. They've all been European. Daniela, I know, is here. She's done lots of work looking at in Spain, looking at puberty methods in Spain. So there's several of the publications that you can explore in your own time. And I've tried to pull together what we know so far about puberty, particularly in the medieval period where we have most of the data. But I have Sasha Valmey and Anders McGovern, two of my PhD students who both graduated, who have looked at Roman and post-medieval samples in the UK. And we're kind of drawing together an example of what's going on with puberty in the past. So the black lines are modern ages of attainment of the different sections, stages of puberty. So you can see initiations around the same, onset of puberty is around the same, slightly delayed for the Romans, and this is the girls. But as we get through to the different stages, you can see that everything starts to lag behind modern individuals. And I'll talk to you a bit about why we can't necessarily compare everything against modern individuals in our data. But the data is there, so percentages of individuals who are hitting each of the points and the ages that they're hitting initiation. So, for example, Menarche here for girls is average age of 14. The Roman period going up to nearly 17, because this is including London girls in the medieval period, and similar to what's going on in the post-medieval period with industrialization. So if you look at the males, we also have some indication of what's going on with male groups. And so Arthur et al. In 2016 looked at some Roman data for males. And we have less data, but still really interesting data comparing what's going on. And again, we have medieval and post-medieval compared to the modern. So the boys are lagging behind the modern individuals, apart from onset, where they're starting slightly earlier than modern boys. And again, we're going on to seeing further delay as you get into the later stages of puberty. So this isn't by any means perfect. It's still just starting to try to pull together some data to look at patterns of puberty in the past. So maybe we can start comparing different populations and saying something about health and stress during that time. So there are lots of issues that we will kind of explore at various times, or you might have questions about as we go through, and things that I've noticed as I'm reading some of these great papers that are coming out about applying these methods. So at the moment, we don't have any known puberty skeletal collections. We can use clinical radiographs of modern individuals where we have puberty data, or for instance, Menarche data. But we can't really test this collection unless we know when the ages that people were actually hitting puberty. We have lots of historical data for when girls achieve Menarche, but we don't necessarily know this is accurate. A lot of it seems very familiar. They kind of stick with between 12 and 14, and with very little variation on theme. So how much that is real and how much they are just recounting what somebody else has said, we don't know. Because you need lots of areas of the skeleton to provide an assessment, you need large sample sizes and well-preserved skeletons to actually look at a population to look at population puberty. But that doesn't mean that when you have one individual, you can't still do a puberty assessment, because you can really pull together a really interesting osteobiography. You have the age of the individual and think about how they would have appeared physically to their population, to their group. When they died, did they look like an adult? Did they still look like a child when they died? There's a lot of interesting things we can explore there. Some of the publications have talked about issues with applying some of the methods. So the method that looks at the neck vertebra, spinal vertebra, and also the wrist bone, the hamate, they can be quite difficult to apply. We'll look at those in the final session. And also Menarche, as you'll find out, most securely relies on the ossification of the iliac crest, which is a tiny slither of bone in the pelvis. And this very rarely survives when it's not ossified or when it's not fusing to the pelvis. So that's a problem in that we have very few individuals that we can actually say they must have just started Menarche at this time. But we explored, Sharon DeWitt and I explored a method of looking at those ages of individuals before puberty and after puberty as a way of comparing different populations and different timings of Menarche. It's less precise, but it still gives you a way into looking at changes that might have occurred. In this case, it was looking at the pandemic, the Black Death, and how that affected Menarche in the past. And also comparing archaeological populations with modern populations is a problem in that we have issues with our aging. We'll talk about next on Thursday. So we have problems with how we age our individuals. There's lots of variation in aging. And we might want to refer to known age collections. So Charlotte Henderson and Padez recently looked at a Portuguese known age collection and had much more refinement in their ages for puberty attainment because they knew the ages of the individuals when they died, even if they didn't know what their puberty status was. So when we're comparing ancient and modern chronologies for different stages, we should expect our ancient populations to be lagging behind modern populations, not necessarily because they're not as fit or they're not hitting targets at the same time, but because of methodological issues of how puberty is reported in the past and how it's reported in modern clinical populations, and also problems with aging standards in that our ages are probably lagging behind modern ages. Age at onset seems to occur very similarly across whatever country or period that you look at, and that concerns me a little bit. There should be variation in onset of puberty. And this might be an artifact of the methods that we use to determine whether someone's just started puberty or not. And we'll talk about that again in the session. But as I said, Henderson and Padez have looked at known age collections, and this allows us to take aging out as a problem when we're using this method. And they have shown earlier ages of onset by using known ages. So this could be a way to exploring onset a little bit more. So finally, I think adolescence and adolescent period is really crucial to understanding what's going on in the past. The new method of looking at puberty can help us track physical development and look at the impact of stress and disease on human development, and then say something about environmental conditions. We have a way into exploring theoretical paradigms such as DOHAD, fetal programming, life course history, and energy trade-offs. And we can also build population as well as individual stories. But lots of challenges in the method still exist, so people need to do more research. It's by no means a perfect method, and all the research is yet to be done. But one of the things I'm trying to do, is quite ambitious, is to collate all of the data that's been collected, particularly for archaeological reports that may be in the grey literature, where people have some data and of an individual, and there's not much more they can do with it. They could send it to Reading, and we will put this data together in a database. All the information about this is on the website for the workshop. And then we can perhaps start building more of these tables and more of these graphs looking at population development. Okay, so before I finish talking, the next session is on Thursday, on the 16th, same time. And we're going to look at aging and sexing of older non-adults. So those of you who are students still at university are probably aware of some of the new methods. Others of you may not have studied bioarchaeology for several years and are unaware of some of the new methods, but also some of the problems that we have with aging and sexing and how that's affecting puberty assessment. So check out the workshop website because there's stuff for you to look at before you come to the next session. And hopefully we can look at some questions and answers of the session. And if you can't get through or can't post anything, then please feel free to email me any questions you might have, and I will post these onto the website as soon as I have a chance to look at them. but they'll be on the website, which will continue on after the workshop. So I'm going to stop talking there, and Kerry, if you can stop recording, that